My name is Rodolphe Barrangou. I'm a professor at NC State, and I've had the great pleasure to be involved in the CRISPR craze and gene editing revolution for 20 years. Gene editing is an amazing technology that's revolutionizing our ability to manipulate DNA, virtually rewriting the code of life in any organism we want, anywhere we want on planet Earth. So in nature, CRISPR-Cas systems provide adaptive immunity in bacteria against invasive genetic elements. So if you're a small bacterium on planet Earth, occasionally you get attacked by plasmids and transposons and viruses of bacteria called phages. And CRISPR in nature is an immune system that allows those bacteria to protect themselves against viruses by having DNA-encoded, RNA-mediated nucleic acid targeting and cleavage. So essentially what CRISPR is in nature is a molecular scalpel that allows bacteria to control an invader genetic sequence and then cut that DNA to destroy it. What's really interesting is not so much what CRISPR does in nature or where it comes from, as much as our ability to repurpose and reprogram that molecular scalpel to cut DNA precisely in any sequence on planet Earth. And when we cut DNA, for example, in human genomes, there's an endogenous DNA repair pathway that patches it back together. And in doing that process, there's a small mutation precisely at the site of cleavage. That's why it's called genome editing, because that small mutation is a SNP, is a change that we make precisely at the site of cleavage in that DNA, and essentially materializes our ability to rewrite the DNA code and rewrite the code of life. So CRISPR as a technology is a molecular scalpel that allows us to rewrite DNA at our discretion. Yeah, genome editing actually existing for decades before CRISPR BC, but it was hard, it was difficult, it was expensive. You needed a lot of resources and a lot of experts to be able to do that, and it took forever. What CRISPR has done is essentially democratize genome editing. Now, the average scientist in the average lab with the average resources can readily, efficiently, affordably, and scalably reprogram CRISPR to edit DNA in any organism of interest to them, whether they want to change DNA in a human cell, in a plant cell, in a tree cell, in a bacterial cell, in a viral entity or capsid. We now have this great tool that has democratized genome editing because it's simple and easy to use. So as a food scientist, I originally came across CRISPR in bacteria that are used to ferment milk into yogurt and cheese. And my team in industry discovered the immune system and the immune function of CRISPR and used that technology to actually make bacteria more sustainable, more functional, and more robust in the fermentation process. And as a matter of fact, products and dairy products derived from CRISPR-enhanced starter cultures that are phage resistant have been on the market globally for over a decade now. Now, since then, that technology has been deployed across the tree of life in therapeutics, in biotechnology, in medicine, in food, in ag, in forestry, and on and on and on. And that technology now is broadly deployed and globally deployed across the tree of life and across industries to engineer and edit genomes of interest, to develop new cures for medicines, to develop new antibiotics for medicines, to develop more resistant or resilient crops and trees on farms, more virus and disease-resistant livestock for breeding, of course, more sustainable trees, and of course, more affordable and more sustainable food supply chain. Yeah, from a scientific and technological standpoint, there's almost no limit to what we can do with CRISPR but our imagination. And we're very creative and very imaginative, so it's not a technological limit whatsoever. I think in the context of societal use and deployment of novel disruptive technologies, we have to be mindful of ethical constraints, right? Are we going too fast? Are we going too slow? Depending on whether you talk to ethicists or patients, there's differences of opinion there. How do we educate a non-scientifically trained audience on the potential, the advantages and the caveats of that technology. And then likewise, one of the major challenges that we're going through right now is because that technology is growing up so fast, so rapidly, so broadly, and so globally, is the need to inform governments and regulatory agencies so they can have science-informed regulations and frameworks. And we're going to get there, but people need to understand there's sometimes a little bit of a lag between the time a technology gets developed and the time a technology gets deregulated and accepted in society. And I think as scientists, it's important for us to not just focus on the science all the time, but also contextualize the work that we do with consumers, with customers, with patients, with farmers, with regulators, with investors, and the public. Yeah, most of you probably don't need a CRISPR-based therapy or a CRISPR-based drug at this point in time in your life, given how healthy and vibrant you are. However, probably unbeknownst to you, I would guarantee or bet that you've all used or consumed a CRISPR-enhanced food product. And as a matter of fact, nearly all fermented milk that turns into yogurt and cheese at the commercial scale has been fermented using a phage-resistant CRISPR-enhanced bacterium, which means if you've had a bite of yogurt, a bite of cheese, pizza, nachos, or cheeseburger, pretty much anywhere in the world last 10 years, I can guarantee you, you have consumed a product that was CRISPR-enhanced unbeknownst to you. I mean, I always love food as a consumer. I always love food as a scientist. It's fun, it's playful, it's engaging. You can touch it, you can feel it, you can taste it, you can play with it, you can try it at the store, you can buy it yourself. So when I was thinking about my career, whether to go into food, to go into ag, to go into biotech, to go into therapeutics, what's fun about food is that there's a new food supply chain every year. There's new products, there's new flavors, there's new technologies, there's new brands, there's new companies, and that enthusiasm and relatability as a consumer and experimentalist and as a scientist to work on food is great and refreshing every day.